Hello friends, in this lecture we will have a guest lecture on a case study on the comparison of methods for bioaerosol sampling and characterization for a low biomass environment. This is a guest lecture by Dr. Akila Muthalagu. Dr. Akila Muthalagu is a postdoctoral researcher at the National Research Center for the Working Environment in Denmark. She specializes in bioaerosol sampling, characterization and analysis with a special interest in the indoor air bioaerosols and has a substantial expertise in these topics. She will present here a case study on a comparison of two sampling methods for characterizing indoor bioaerosols in a low biomass environment.
Hello everyone, welcome to another lecture on indoor air pollution monitoring. In today's lecture, we are going to see a case study on comparison of two methods for bioaerosol sampling and characterization in a low biomass chamber environment. Before we jump into the case study, I want to give a small background on why is it important to see the efficiency of bioaerosol samples. So we all know that bioaerosols such as bacteria, fungi, viruses are very much abundant in the environment. Nowadays, there are even studies which showed that there are few species of bacteria in the outer space. So we know they are very much abundant in the environment and across various fields. But they have a unique nature. They can have both positive and negative effects. For example, there are studies which showed that early exposure to environmental microbes have a positive influence on immune system and reduces the risk of asthma in later life, meaning that when we as a kid when we exposed to various microbes, we acquire certain immunity. Similarly, they can also have a negative impact. For example, presence of Streptococcus pneumoniae or the bacterium called Legionella can have a um, negative impact on human health. It can cause fever or it can cause respiratory related issues. So they have both effects. So it's really important to understand what is there in the environment where we live, where we spend most of our time. So when it comes to understanding the microbial communities in the air, bioaerosol samplers plays a vital role because they are the one which is helping us to collect the sample and understand the diversity and richness in a particular area. Because of this interest, because of this developing interest on bioaerosols, there are diverse array of samplers in the market. You can get there are a lot of different samplers that are available. Each sampler offers unique capabilities, and their application for capturing airborne microorganisms could vary. So it's important to explore these samplers, and we should understand how their efficiency, sampling efficiency changes based on the environment. Why is it important? Because bioaerosol sampling, when we talk about sampling and analysis in microbiology, in aeromicrobiology, especially in bioaerosol sampling, so far there is no standard protocol or there is no one single recommended sampler available for sampling. So it's really tricky to choose which sampler to use or which analytical technique to do to characterize the bioaerosols. So when we talk about sampling in the bioaerosol field, there are two types of sampling. One is active sampling, another one is passive sampling. Both the samplings are sampling types are used to characterize the air microbiome. So active sampling involves actively drawing air samples using a pump or vacuum. Whereas for passive sampling, it majorly relies on the natural process like diffusion or settling. So both the sampling techniques have major advantages and disadvantages. For example, if we look into active sampling, the major advantage here is like this sampling provides a control. We have a control and precision over the sampling technique, because we can set the sampling volume, how much air we want to draw, and for how long, for whether it can be 2 minutes, it can be 2 hours, it can be 24 hours. That's up to us. And because of this particular reason, this sampling can quantify how much bacteria is there in the air, and also qualitatively, we can also do qualitative results, meaning that we could count and we also could be able to tell what is the different species present. But whereas if you look at the disadvantage on active sampling, there are different types of samplers. So the cost of the sampler can be high, and also these samplers required calibrations, which is again a cost related issue. And another thing is, this active sampling requires power source for operation. So in some places it's not possible that we have a active power source, and so meaning that we cannot use this sampling everywhere. And another important drawback here is, like, since we are drawing the air, we might disturb the microbial balance in a particular environment. So when we look into passive sampling, the advantages are, you really don't have to spend much money on samplers. You can use a settle plates and or open petri dishes. We can keep it on a desirable place where we wanted to do sampling. We can leave it for longer period, and then we can take the settled particles and do the analysis. If we don't need any external power for this particular sampling. This since the sampling time is long, it offers a realistic microbial distribution representation, and it won't disturb the microbial balance of the environment. But one major disadvantage of this technique is, since we don't have control over the sampling process, we cannot do the quantitative analysis, meaning we don't know we won't be able to tell how much of the concentration is there per meter cube of air. That is not possible to tell here, but this technique can be used for qualitative alone. Second disadvantage of passive sampling is, we don't have we have less control over sampling parameters, because this sampling majorly depends on the settling process. So if wind is high, we might not get enough material on the plate. If we have higher relative humidity, because of the moisture, many particles might settle down. Again, that might not be the scenario in another sampling place. So we really don't have control over these parameters, which could influence the sampling amount which we get. So because of this disadvantages, active sampling is the one which is widely used for bioaerosol sampling. So when we talk about active sampling, as I mentioned earlier, there are numerous amount of samplers available. However, all of them comes majorly under four categories, which is called impingers, cyclones, impactors, and filter samplers. So for impinger and cyclone, the usually sampling medium is liquid, and for the impactor, it can be either solid or semi-solid mediums. And for filters, we have different types of filters on which the PM material will be collected. So again, if we look at the figure here, I have there are 4 different samplers here, and each of them represents each category. The first one here, it's a biosampler from a brand called SKC. So what you do is, like, the down you can pour the liquid of your interest. Mostly we use saline, and if it is for virus sampling, there are some solutions. So basically the medium can vary based on the interest. So we pour solution here, and the pump is connected, and we can set the sample volume as per the manufacturer instruction, and we can do the sampling as per our aim and objective. For example, if you are going to do sampling at 14 lpm, I mean 14 liters per minute for 30 minutes, the air is gonna draw here, and all the particles will get collected on the solution here. And this one, the second one, is a Coriolis sampler. It's a cyclone sampler. So nowadays there are different types of cyclone sampler, and there are also different type of impingers. I have shown only 4 examples here. So again, this is a Coriolis sampler. In cyclone sampler, you can there can be a filter paper inside, or there can be a medium inside. So for example, in this sampler, let's say I am going to do bacteria, then I can choose my own medium. Again, that's a question based uh, research question based one. For example, if I'm looking for bacteria, I can go with tryptic soy agar, or nutrient agar, or mechaniki agar. It's of my interest. Like it specifically which microbes I want to go. Based on that, I can choose my medium. Similarly, if I want to go for fungi, I can choose a potato dextrose agar, or TG18 agar, or MY50. It totally depends on my objective. So I can put the plate here and turn it on. It's gonna collect the air, and all the spores and microbes are gonna settle down on the medium inside, which I can incubate later and do the further analysis. The third one is called SKC Biostage Impactor. It's a single stage sampler. There are also 6 stage on this, 3 stage on this. There are different type of samplers. So the idea here is also same. It can be worked with both filter and also with different media. So like I mentioned about cyclone sampler, you can also do the same here. You can use different media, connect the pump, set the flow volume, and do the sampling for as much as you want. And the last one is a button sampler. So this is, I think, for 37 filter paper, but there are samplers with 47 also. And filter paper, again, it's a research question. So some people use PTFE, some people use polycarbonate, some people use quartz microfiber. So the efficiency again changes depends on what type of filter you are using. That's another question. But here we are talking only about the samplers. So this is a filter based sampler, and here you see this small mesh kind of thing in which the air will get collected, and it will the filter will collect all the particles on it. So these are the 4 different samplers which we could use. Again, one important thing to notice here is, like, these samplers sometimes comes with a cutoff value. It can be for PM 2.5 or it can be for PM 10. So again, it totally depends on our objective. If you want to have a long spectra, long size spectrum, we can go with PM 10. Or if we want to have majorly we are looking for a smaller size, then we can go for PM 2.5. So these are some of the samplers that are widely used in the market. So when we look into the literature, there are a lot of studies that focuses on the biosampler efficiency. Why? Because, as I mentioned earlier, there is no standard protocol for bioaerosol sampling, and there is no standard analytical method. So there are many studies which involves in understanding how this sampler works in different environment. But the problem here with in many of the studies is these studies either use culturing technique or they target a specific microbe to check the sampling efficiency. Why it's a problem? Because when when you use a culturing technique, we should note that not all the microbes in the air are culturable. There are more than 50% of the microbes in the air that cannot be cultured on a medium in a lab, or they need a very specific conditions. So when we do efficiency tests through culturing techniques, these might not represent the original scenario. That might not be the original efficiency of the sampler. Similarly, when you target a specific microbes, for example, I chose only Bacillus anthracis, and I'm checking three different samplers, and I wanted to know how they are able to capture the Bacillus anthracis spores. But the problem here is they might work very well for Bacillus anthracis, but they might not work well for the Pseudomonas species. So when you target a specific microbes and say it works well, that might work well for the particular microbe, might not work well for another microbe. So the efficiency could vary depending on the microbial communities as well. And another thing, there are when they do the targeting, many of the studies are done in a laboratory scale, meaning that they had this artificial bioaerosol generator in which they generate the microbes and they try to capture. Again, the problem here is when you do in a lab scale, you skip a lot of other environmental factors which could influence. For example, relative temperature, relative humidity and temperature, wind speed, all these factors could play an important role in the microbial composition and how the sampler is collecting those. When we do a laboratory analysis, these things might have skipped, and our efficiency might not be the original one. Similarly, when we do these analysis under different conditions, I mean in the outdoor scale, that also a problem, because when I do sampling in the outdoor environment and I want to analyze the efficiency, there are a lot of factors on which I don't have control over. For example, again, wind speed. If when I do the sampling, if wind speed is so much, it could disturb the microbial composition, and the concentration on the particular area might be less because microbe moved away. So the sampler might not work well at that situation. So the efficiency, so I might say that it's not efficient for microbial composition, but that's not true. If wind speed is not that much, then microbial composition might not have got disturbed, and I would have got a different results. So these are the factors that are really important to consider when you check the efficiency of the sampler. For example, if you look into the studies, there was a study in 2019 which showed that electrostatic precipitator samplers had higher collection efficiency compared to the membrane filter or liquid impincher sampler in an outdoor environment. And again, another study stated that higher microbial diversity with the filter samplers compared to the wet cyclone sampler in real life environment. So this is what I was trying to tell. When you do in the outdoor environment, we don't have control over the uh, environmental factors, so results could really vary between place to place. It's also so it's important to note that these factors have influence on the microbial behavior and sampler efficiency. For example, there are studies which showed that uh, in a farming area, the efficient in a swine box, liquid impingers and filtration based samplers worked very well compared to the Anderson sampler, because if you take any cattle field, there are a lot of sources. For example, the presence of pets itself could act as a source. Then if the place has high occupancy, or whether they have any water leakage or any air conditioner usage effect, these are the factors that could influence the bioaerosol concentration. So when you do even in the indoor, these we have to consider these factors as well. So outdoor alone is not a problem. Even in indoor, when we do the sampling testing, these you have to really consider that some houses, like a house with two people, are not equivalent to a cattle field, because they are rich in sources. So there is a very much limited knowledge existing regarding the efficiency of sampler. Um, uh, in indoor also, if we check, the compared to the outdoor environment, indoor environment is considered as a low biomass environment. When I say indoor environment, I am not talking about the places which is rich in diversity or rich in sources. I'm talking about a normal, let's take a normal household with the two people. It's a normal indoor environment with the low biomass. So if I want to go to house and do some sampling, that might not represent the other scenarios. So it is really important to know how much efficient a sampler is in such a low biomass environment conditions. So by considering all these things, we kept our objective that we will test the performance of a sampler under a low biomass environment. And another objective is to see how the microbial concentration and composition varied in with these two samplers, and what are all the bias that are introduced by the samplers. So to control the environmental factors, we chose to do the study in an environmental chamber. So this is the figure which shows this is a schematic figure of the environmental chamber where we did the study. Why we chose the environmental factor is, as I told earlier, temperature, relative humidity, wind, all of them have a huge impact on samplers efficiency. So to control those factors, we chose the chamber. So this is a stainless steel chamber. It has two chambers, but our study was done in the first one. So if you see here, the outdoor air is introduced through downs through the base of the chamber, and it was exhausted to the upper ceiling. So you could ask me, okay, so you are introducing outdoor air only, right? So it might introduce some outdoor bioaerosols. So how can you say it's not affecting? Here what we did to avoid that scenario is, the air which is introduced from the outdoor is filtered in 4 different stages. So we made sure that nothing comes from the outdoor. I'm not saying it's 100% clean air, it might be 99.9%. So the outdoor air is introduced through this platform, and it comes to the indoor and it goes away. So since our interest is a normal indoor environment, we put 4 people inside. So here people are the only sources. So compared to cattle field or any other hospital environment, houses have very limited sources when only humans are there. So we considered this as a low biomass environment. So there were 4 people inside, and we put 2 fans. So the air is uniformly mixed, so the contribution so the composition all over the place will be similar, and the temperature, relative humidity was monitored throughout the experiment, and we were also measuring the CO2. So this is the chamber. In the next figure, I will show. This is the indoor picture of the inside picture of the SAM chamber. Sorry. So here I am showing the two samplers which we used. So the reason for choosing these two sampler is these are majorly used samplers in the field. One is based on the filter, and another one is called cyclone sampler. The difference between these two sampler is this cannot the filter sampler, which will be mentioned as personal environmental monitor PEM in later slides, it cannot size differentiate, whereas the cyclone sampler can size differentiate. For example, the first stage collects more than 4 microns micrometers of particles. The second stage collects 1 to 4, and the third stage collects less than 1 micron. So the both of them are dry samplers, and both of them have different sampling volumes, which we will discuss later. So if you see, we had like 4 people, meaning that two, I mean, in both the corners we had two tables, and people were sitting here. And if you see, this is how we installed the PEM sampler. So this is here, and this is the cyclone sampler we installed at in the middle. The reason for having so many PEM sampler and only one cyclone sampler is because they have different sampling volumes. The PEM sampler works at 4 liter per minute, but cyclone sampler can work at maximum 10 liter per minute. So to compensate that, we had multiple PEM samplers and only one cyclone sampler. As I told, we had fans, which is why we considered this as a well mixed chamber. And the reason to keep at this height is because we can have it at a breathing zone level. That's why it has been installed here. So this is the total sampler setup, and this is from SKC, and this is a replicated version of a NEOS cyclone sampler. So both of them are recommended samplers, and we wanted to see how these samplers perform in a low biomass environment. So coming to the analysis, as I mentioned, the sampling was done using two different samplers. We did DNA extraction for both the samplers. The DNA extraction was followed by DNA quantification. We checked how much DNA is there in both the samplers. Then we did qPCR. It's a quantification technique. So we targeted the 16S rRNA for bacteria. Fungi is not focused here because humans emit more bacteria compared to fungi. Since it's a low biomass environment, we wanted to see how the bacterial concentration is. This study is a continuation of previous study. So in previous study, we found that fungal concentration is way too less compared to bacteria. That is the reason that we chose only bacteria here. So the concentration was calculated based on qPCR. After concentration, we wanted to see how gram positive and gram negative bacteria concentration is in and what's the change in concentration between the samples. So this is quantification technique. So when we then we also did qualitative analysis. For that, we chose NGS because NGS is next generation sequencing, which is a advanced level. Why we chose this? Because as I mentioned earlier, not all the microbes are culturable. So we wanted to have a better comparison. Excuse me. For that reason only we chose NGS. So NGS is a sequencing technique which can which can identify both viable and non-viable bacteria. So for NGS, after the DNA extraction, we prepared the library based on the manufacturer instruction, and we did NGS in a Illumina platform. After NGS, the data was analyzed in R for richness and abundance calculation. Then we compared both these results between the samples and among the samplers, and then we finally calculated how much bias was introduced uh, between by the samplers in terms of results. So this is a schematic diagram which shows how the all the analysis was carried out. As I told, we had PEM sampler, we had multiple PEM samplers so that we can compensate with the cyclone. Cyclone, the average, it can go up to 10 liter per minute, but at the end of sampling, the average was 7.9 lpm, whereas PEM worked at 4 lpm. So we had 6 PEM samplers and 2 stages in the cyclone. Since PEM cannot do size distribution, we have combined these two together. So how we did is, it's a dry sample. Both of them are dry sampler, but in PEM we used polycarbonate filters to collect the samples. So those filters were immediately stored in the extraction kit's first solution. It's called CD solution. We used a kit called Qiagen Power Soil DNA Extraction Kit. In that, there is a first stage solution in which we stored the filter directly. And for the cyclone, since it's two different tubes, one is, the first stage is 15 ml tube and another one is 1.5 ml tube. So we have added CD solution to each tubes. We mixed it well, and then we pooled that solution together so that we don't have to do the size distribution. We combined it together so we can have a fair comparison. Similarly, for all the 6 samplers were pooled together and DNA was extracted. So at the end of the day, we had only one sampler per day. So we had 11 samplers, 11 samples for each sampler, and those samples were collected parallelly. So we can have a proper comparison. So after DNA extraction, DNA was quantified, qPCR was done. qPCR was done in terms of total bacterial concentration, gram quantification, gram positive quantification, and gram negative quantification. For NGS, we did alpha diversity analysis and beta diversity analysis. At the end, we compared everything and we saw which sampler had better performance. So this is the result which shows the DNA quantification. So the blue one represents how much DNA was obtained from cyclone sampler, and the red one shows how much was from the PEM sampler. So we could see a clear trend here. As I mentioned that we had 11 days of sampling. So we could see in all the days, DNA concentration was consistently high in the cyclone sampler, and it was significantly high in all the sampling areas. So what could be a reason for lesser DNA concentration in PEM sampler? So one reason could be the PEM sampler works at though it works at 4 liter per minute, it collects the particles on the filter paper. So there is a good chance that due to desiccation, because it's a dry sampler, due to desiccation, some microbes would have been dead, or some particles would have been bounced back. That could be a reason that we have less DNA concentration. Then what next we did is we did qPCR. As I mentioned, we have targeted the universal 16S rRNA gene for bacterial concentration. So this is the total bacterial concentration between these two samples. Again, the blue represents cyclone and red color represents the PEM sampler. If you see here, in similar to the DNA quantification, we could see all the sampling days for cyclone sampler was significantly high compared to the PEM sampler. If we see for the cyclone, the maximum was almost close to 3 into 10 to the power 6, whereas for PEM it was 2.2 or close to 2.5 into 10 to the power 6. So we can see that there is a huge variation in the sampling days itself. And the trend was consistent among all the sampling days, and it's also significant. So, as I mentioned, lower concentration could be due to the desiccation and particle bouncing. Another issue to notice here is when we use a filter, what type of filter are we using is also important. This is what I mentioned in my earlier slides also. Some filters, we are collecting particle on the filters. So when we do the DNA extraction, it's really important that we recover all the particles from the filter paper. If something, if the filter is not desiccated properly, or if some particles are stick to the filter, we might lose the particles, and we might underrepresent the uh, uh, results. This could be one of the reason. But so far, cyclone sampler works very good for quantification compared to PEM. So the next idea was to see, to differentiate the sampler's performance in terms of gram positive and gram negative analysis. Here, if you see, this one is for gram positive, and this one is for gram negative. So here you can see there is no proper trend for gram positive bacteria. If you see here, the PEM sampler had little higher, and if you see in these two samples, PEM sampler had huge difference compared to the cyclone sample. But again, if we see on this last 4 days, the cyclone sampler had higher concentration compared to the PEM sampler. So it's really difficult to say which sampler is better than the other one. So to know that, we did some statistical test. We checked if there is any significant difference in the concentration by using a Kruskal-Wallis test, and we could see the P was more than 0.05, which is showing that there is no significant difference in the concentration between the sampler. So what could be the reason for this? Is that the gram positive has a thicker cell wall, so it might not desiccate that easily, or it might not die that easily. That could be a reason that both the samplers worked well for gram bacterial concentration. So the result from this, the conclusion from this graph would be like both the sampler works better, had both the sampler had similar performance for gram positive bacteria. But if we look into the gram negative bacteria, we could see that there is a proper trend. In all the 11 sampling days, the cyclone sampler had huge concentration compared to the PEM sampler. So you could ask me that, okay, the cyclone sampler worked at 10 liter per minute, and the another one worked only for 4 liter per minute. So that could be a reason. So if you notice here in the axis title, it says genomic copies per meter cube. So all the graphs and all the values here are normalized based on the sampling volume. So when we get the results, so the conclusion from here would be that when we want to quantify the gram negative bacteria, cyclone sampler would be a better option than the PEM sampler, because they have a better efficiency for gram negative bacteria. The reason could be, as I mentioned earlier, compared to gram positive bacteria, gram negative bacteria has a thin cell wall, so they easily they are vulnerable to desiccation. So since it's a dry sampler, that could be a reason that we have lesser concentration in the sampler. So now we have done the quantification, and we, based on the quantification, we can conclude that okay, so cyclone sampler had a better performance than the PEM sampler. So now next what we wanted to do is we wanted to see how the samplers works for the composition. So our idea is not only to know how much concentration is there, but our idea is to see what are all the different species are there. So for that we have to analyze the sample, and that's the reason we did NGS analysis. So when we wanted to see the composition, the first thing we did is we opted for few statistical tests which can tell us which sampler had better diversity. So for the in Bioaerosol studies, the important statistical analysis which we do is using different diversity indices. We use different diversity indices. So there is two common diversity indexes, which is called alpha diversity and beta diversity. So in alpha diversity, there are many indices which can be used. I used Shannon and Chow 1. So they tell how rich, how diverse the environment and how rich an environment is. So if we look into the first figure, this is the alpha. So the y-axis represents how much is the richness indexes, and the x-axis shows the sampler names. If you see the diversity indices for the PEM sampler, for SHAO 1, it is high. If you see the median, it's more than 450. But if you see here for cyclone, it's closer to 375. So there is a variation in abundance. Like there are more number of species in PEM sampler compared to the there is a the species are highly abundant in the PEM sampler compared to the cyclone sampler. So the next question we wanted to see is like how diverse that environment is. For that we used Shannon. If you see here, again similar to the another other diversity index, here also the trend is same. You can see there are the Shannon index is close to 4.5 for PEM sampler, and for the cyclone sampler it's less than 3.25. So which shows that the PEM sampler alpha diversity is highly the PEM sampler has highly rich and diverse microbial composition compared to the cyclone sampler. This is very much contradictory to the previous quantification results, but this is what we got. So next what we did is we wanted to see, okay, we know now like there is a difference in richness and abundance between the samplers, but we wanted to see how whether there is a significant difference. So for this alpha diversity indices also we did Kruskal-Wallis to see whether they are significantly different, and yes, we could see there is a significant difference between these parameters and among the samples. For the next one, we did beta diversity by using PCOA analysis. So we could see that all there are two planes here, and the samples from PEM lies in one plane, and the samples from cyclone lies in another plane. So we could visibly see there is a significant, excuse me, so the samples lies in two different places, and all the samples are clustered together. For example, if you see PEM samples, all of them are together here, and the cyclone samples, they are together in another side. So from this visually we can say that okay, they are different from each other. So how to prove that statistically? For that we did a test called ANOSIM, which is called analysis of similarities or dissimilarities. So we could see that that test also showed that there is a significant difference between these two sampling events, microbial composition of these two samplers. So the diversity indices showed so far that when it comes to richness and diverse, PEM sampler has upper hand, and both the microbial composition in both the samplers are significantly different. So what would be a better way to look into this? So what we next did is we looked into the microbial composition. So this is the graph which shows how dominant. This shows the top 5 dominant phyla from both the samplers. If you look into the bar here, we could see that the composition again is very much different in both the samplers. For example, if you look at the cyclone sampler, the actinobacteria is almost represents 40%, followed by formicutes, which again is close to 40%, followed by proteobacteria, which contributes for the another 15%, and then remaining is bacteriota, and other few are compiled as others. But if we look at the PEM sampler composition, we could see the majority of the microbes comes from the proteobacteria, followed by actinobacteria and firmicutes. So we could see that the cyclone sampler is dominated by the two phyla called formicutes and actinobacteriata, whereas the PEM sampler is majorly dominated by proteobacteria. So again, we did a Kruskal-Wallis test. We know it looks different, but whether it's statistically different, to do that, we did a Kruskal-Wallis test, which showed, yes, they are significantly different from each other. So, one important thing to notice here is from our quantification, we saw that cyclone sampler had upper hand for the gram-negative bacteria, whereas PEM sampler did not do well. But the thing to notice here is majority of the proteobacteria are gram-negative bacteria. So when you see the higher relative abundance of proteobacteria here, which is contradictory to the previous results. So it might not work well for quantification, but it was able to capture a lot of species. So one thing to note here when we talk about quantification, we did qPCR, which is genomic, so the concentration here represented in terms of genomic copies. So genomic copies can vary from species to species. For example, if I have bacilli and if I have a staphylococci, bacilli can have 10 gene copies and staphylococci can have 2 gene copies. So when I have 2 bacilli and only one, even if I have 5 staphylococci, bacilli will have higher genomic copies because they have 10 with them. So it's 2 of them will contribute to 20 genomic copies, whereas 5 staphylococci can contribute to only 5 genomic copies. So this is one important thing we should remember whenever we quantify. So that the quantification with qPCR might be different, might not be the original concentration, because here the concentration is represented in terms of genomic copies. And another thing is, here the major we found is Pseudomonas. So Pseudomonas has been reported that Pseudomonas has high adherence to falcon tubes. Why I'm mentioning here? Because if you remember the structure of the cyclone sampler, it has 2 falcon tubes where the samples get collected. So if it has high adherence, it might be difficult to remove them from the tubes itself. So it might not be removed from the tubes for further identification. So we might have missed out there. So that also could be a reason. And so after looking into phyla, we wanted to see genus level, how these both samplers had, how many different genuses were there, and how much, which one was dominant. So this is the this is the figure which shows the top 10 abundant genuses of cyclone samples. Here we could see that the cyclone sampler is dominated by Staphylococcus, the genus Staphylococcus, followed by Corynebacterium and Cutibacterium. So Staphylococcus contributes almost to 25%, followed by 15% of Corynebacterium and close to 12% of Cutibacterium. So one thing to notice is majority of the Staphylococcus are like gram positives, and many of the species here in this list are gram positive. This could be a reason why we have high abundance of gram positive bacteria in the cyclone sampler. If we look into the PEM sampler, we could see that there is no major abundant one here, because as I showed in my previous slides, the PEM samplers are rich and diverse, meaning that they have lot of microbes which could contribute, which might not contribute to a larger portion, but they can, but there are a lot of them. For example, if you remember the Chow index, there were Chow indices for PEM sampler was more than 450, but for the cyclone sample it was less than 350. So it shows that the PEM sampler has more rich and diverse species. So that could be a reason that when we plotted top 10, the majority of them comes in other section, meaning that there are lot of there are lot of species which contributes to smaller fraction, and when we put all of them together, that comes close to 47%. So there are a lot of small species which are present that contributed to 47%. But if we wanted to see what is the one dominant species, then we could consider Pseudomonas as a dominant species, because that contributes close to 16%, I guess, followed by Corynebacterium and Chivanella. So, as I mentioned earlier, this is a gram-negative bacteria, and there are a lot of gram-negative bacteria in the other section as well. And this could be a reason that we have higher gram-negative bacteria. We have proteobacteria as a dominant one in the PEM samples. So what's the next thing which, so far we were seeing, we have seen quantification, and we have also seen the composition. So now we know that we have two different results. For example, okay, from quantification we concluded that the cyclone sampler had better results, but when we looked into the composition analysis, then we have different results. We see that PEM sampler have more diverse and richness than the cyclone sampler, and it has a different, the composition of the bacteria is really different, and we were able to see that even the top 10 genuses are different between the samplers. So what would be the next step? So now we want to see, okay, both the samplers have different results. How much bias are they introducing? That for to do that, we did this passing Poblack analysis and plant Altman analysis. So this test will help us to understand whether two samplers can be used parallelly, or which one works better for a particular scenario. So we did this for gram positive as well as gram negative. So here we could see this is gram positive. This is the result of gram positive. If we see here, this is the result from both the samplers. So the x-axis is from gram positive concentration from PEM sampler, and the y-axis is from the cyclone sampler. So there is a this is the passing Bobluck fit, and we see our samplers lies in the line, very close to the line, meaning that these two sampler have similar performance. If the performance is not good, we will be having this dots all over the place, which will show that they have different performance. So I mean, having these dots very close to the this line shows that these both samplers can be used, you know uh, they both can be used for positive bacterial concentration. This analysis shows there is no proportional and systematic differences introduced by both the samplers. So to confirm it, so now we know these two samplers can be used interchangeably. But when I did for gram negative bacteria, as I mentioned, these dots were all over the place, which shows that they have different performance. So they do have a systematic and proportional difference. So the next thing we did is we wanted to see how much bias is introduced. Again, we did the same thing. We did for gram positive as well as gram negative. So this is a bland Altman analysis results. So the x-axis here is the average concentration of cyclone and PEM, and the y-axis shows difference in concentration of cyclone and PEM sampler. So here we have this 95% limit. This is the maximum and minimum line, and we have a mean line. Again, we have dots very close and within these two lines. If this shows that there is the samplers did not introduce any bias to the results, meaning that they both can be used interchangeably for gram positive quantification. When I did for the gram negative, these stats were above these lines, meaning that they are having different results. So this results were very much inconsistent with our qPCR results, that both the samplers have same performance for gram positive quantification, and they cannot be used interchangeably for gram negative quantification. So the outcome and the overall outcome from this study and the conclusion is, the both samplers worked for a low biomass environment. For bacterial quantification, for DNA quantification and total bacterial quantification, cyclone sampler had better efficiency. For gram positive bacterial quantification, both the samplers can be used interchangeably because they have a same performance, and they did not have any systematic or proportional difference. Neither they introduce any bias to the sampling. But for gram negative quantification, cyclone had upper hand. When it comes to microbial composition analysis, PEM sampler works better for proteobacteria, because if you remember the first plot, you could see that PEM had more than 40 percentage of proteobacteria in it, but very less than 20% of actinobacteria. Whereas if you looked into cyclone sampler was dominated by actinobacteria and formicutes, which shows that this cyclone sampler could be used could might work well for actinobacteria, and formicutes might not work well for proteobacteria. So the overall conclusion from this study could be, when we want to choose the sampler, the choice of sampler is highly depend on our research question. For example, if I want to go for quantification, cyclone might be a better choice if I want, because it had really good results in quantification. But if my research objective is to see only the microbial composition, or if I want to see only gram negative bacteria, PEM might work well. And or if my research objective is only to see gram positive bacteria, cyclone might work. Similarly, if I want to focus only on staphylococci, cyclone sampler might be a good option. Or if I want to look for pseudomonas, my research objective is to look into different pseudomonas, different species of pseudomonas, then PEM sampler might be a good option. So choosing the sampler is highly depends on the research question which we have. And also there are a lot of factors, and there are a lot of different things we have to remember when we choose the analysis. For example, if like I mentioned earlier, even choosing the filter paper, we have to be very careful which filter paper we are choosing, because each filter paper has different efficiency in sampling collection. And when we analyze that filter paper, again we have to be really careful in to digest whole filter, or we have to make sure that we collect every particle from that filter. So choosing the technique and choosing the sampling for indoor air pollution monitoring is highly research question based. That's what I would like to conclude from this case study. Thank you.